Hello and welcome to this Endocannabinoid System training. Today we'll explore the clinical and pharmacological foundations of the endocannabinoid system. As well, we'll explore its role across various physiological systems and the therapeutic strategies that have emerged as our understanding of its mechanisms has evolved. The purpose of today's training is to help bring some much needed context and clarity to your understanding of the endocannabinoid system. Today, we'll be exploring the endocannabinoid system and its role in health and disease, and its growing relevance in modern medicine. Let's begin with how this remarkable system was discovered and why it has reshaped our understanding of homeostasis in the human body. Before we dive in, a quick introduction. My name is Chris, and I'm the founder and CEO of Global Cannabinoid Solutions. We're a science first consultancy dedicated to advancing cannabinoid education and innovation. My mission is to bridge the gap between cutting edge science and real world application, bringing evidence based insights to healthcare, policy, and business. The discovery of the endocannabinoid system marked a tremendous turning point in our understanding of how cannabinoids affect the body. As Roger Pertwee describes, it expanded the focus of research beyond plant based cannabinoids like THC and CBD to include the body's own endocannabinoids such as Anandamide and 2-AG. Today, researchers not only study how these compounds bind to receptors, but also what physiological events trigger their release and how they're taken up and broken down, and critically, how this system influences health and disease. The ECS was discovered through the research into the effects of delta 9 tetrahydrocannabinol, aka THC, the primary psychoactive compound in cannabis. It wasn't until 1992 that Raphael Mechoulam and his colleagues isolated Anandamide, the first identified endocannabinoid, followed by the discovery of 2-arachidonyl glycerol, or 2-AG. These findings revealed an endogenous cannabinoid signaling system present in humans and other animals. To understand the medical potential of cannabis, we need to first go back in time to when scientists first started asking, how does THC actually work in the body? That question led to one of the most important discoveries in neuropharmacology, the endocannabinoid system. What started with a plant compound, THC, ended up revealing a powerful system that our own bodies rely on to maintain balance. And that's why today the ECS is at the center of so much medical research, from chronic pain and anxiety to autoimmune conditions and neurodegenerative diseases. Now that we've seen how the endocannabinoid system was discovered, we can begin to understand why the system matters so much in modern medicine. These discoveries weren't just academic milestones, they opened the door to a new era of scientific research focused on how we can clinically target the endocannabinoid system to treat real people with real conditions. The clinical trials in this slide demonstrate the therapeutic efficacy of cannabinoids in managing various conditions by modulating the endocannabinoid system. This is precisely why understanding and managing the system is so crucial to improving patients' quality of life. The endocannabinoid system, or ECS for short, is not just a collection of receptors and ligands, it's a complex lipid-based signaling system that plays a central role in regulating physiological balance across multiple systems in the body. One of the key functions is the modulation of neurotransmission. By controlling the release of neurotransmitters, the ECS helps regulate mood. It's also deeply involved in inflammatory regulation. Through the activation of CB2 receptors in immune cells, the ECS can either promote or suppress inflammatory responses, depending on the context. This makes it a powerful target for diseases characterized by chronic inflammation. As well as this, the ECS is integral to the reproductive processes and has been investigated for therapeutic targeting in males and females to promote fertility. In the context of energy metabolism, the ECS also helps regulate appetite, energy storage, and expenditure, which is why it's linked to obesity and metabolic disorders when dysregulated. At its core, the ECS is a system of homeostatic control. It responds dynamically to physiological changes, working to maintain internal stability despite external or internal stressors. And that's why the ECS is not only fascinating from a biological standpoint, but also from a therapeutic perspective. Now, here's a little insight that transforms our understanding of human physiology. Endocannabinoid signaling has been identified in all tissues, organs, and bodily fluids studied so far. The endocannabinoid system interacts with every single one of these systems. This positions the ECS not as a secondary or isolated network, but as a master regulatory system, one that is intrinsically involved in maintaining homeostasis or biological balance across the entire body. When we say health, we're talking about the dynamic balance across these systems, and the ECS is right at the center of that conversation. So, what exactly is the endocannabinoid system made of? Well, the ECS is a biological system composed of three key components: endocannabinoids, receptors, and the enzymes that regulate them. These components exist in dynamic proportions, and their balance is essential for maintaining homeostasis, as we've mentioned, or stable internal functions virtually in every physiological system we've discussed and researched thus far. But here's where it gets even more interesting. The composition and activity of the ECS is not fixed. It varies from person to person and is influenced by factors like age, weight, sex, and overall health status. And when the system becomes dysregulated, when these dynamic elements fall out of balance, we often see correlated faults in disease states. Conditions such as inflammation, anxiety, metabolic disorders, and neurodegeneration all have shown links to altered ECS function in the scientific literature. This isn't just a passive system, it's a responsive, adaptive network designed to fine tune the body's stress responses, immune tone, neurological signaling, and much more. So, as you can see, understanding these components and how they shift under stress or disease is critical if we want to use cannabis based or cannabinoid targeting therapies strategically and effectively. And so, what exactly does the endocannabinoid system look like at a cellular level? And so, one important thing I want to point out is that the ECS operates through a very unique mechanism in the brain known as retrograde signaling, something we'll keep touching on later. Now, this means that unlike most neurotransmitters, which travel from presynaptic to postsynaptic neuron, endocannabinoids are synthesized on demand in the postsynaptic neuron, and then they travel backwards across the synapse to the presynaptic neuron. So if we break that down slightly, step by step, when a neuron becomes overactivated, for example, due to excessive glutamate release, enzymes in the postsynaptic cell rapidly produce 2-AG or anandamide, AEA, from membrane lipids with the support of these synthesizing enzymes. These molecules then diffuse retrogradely into the synaptic cleft through the endocannabinoid membrane transporters, or EMTs, and they then bind to CB1 receptors located on the presynaptic terminal. And so, once activated, these CB1 receptors inhibit further release of neurotransmitters, acting as a bit of a brake system in the neuronal messaging process. And it's this that allows the neuron to self-regulate and restore balance and maintain homeostasis. And so after exerting their effects, the endocannabinoids are then taken up and degraded by their respective enzymes. So FAAH, FAAH, breaks down anandamide inside the postsynaptic neuron, whereas MAGL, which degrades 2-AG, does the degradation process in the presynaptic terminal. So this entire feedback loop is crucial to maintaining synaptic plasticity and it overall helps regulate processes like pain, emotion, appetite, and memory. So while both anandamide and 2-AG are classified as endocannabinoids, their roles within the ECS are very distinct but highly complementary. And 2-AG is typically present in higher concentrations, and it's more often involved in broad, rapid neuromodulation, often recruited in response to, say, acute stressors or injury. Anandamide, by contrast, appears to play a more tonic or regulatory role, often involved in fine-tuning synaptic activity, mood modulation, and subtle shifts in behavior or perception. So the emerging research is starting to suggest that the temporal and spatial coordination of these two molecules creates a bit of a layered signaling mechanism. So 2-AG delivers the initial push, and then anandamide modulates the aftermath. And so this layered response allows for a more precise physiological adaptation, whether managing immune activation, modulating pain perception, or recalibrating emotional tone. And so, importantly, their synergy extends beyond receptor activity. Their metabolic pathways, transport proteins, and interactions with non cannabinoid targets all contribute to this wider, broader regulatory function within the network, which hints at a number of different roles in everything from appetite signaling through to neurodevelopment. So, how does something like stress affect the levels of these endocannabinoids in the body? Well, when the brain experiences acute stress, the first molecular change is a rapid drop in anandamide levels in the amygdala. This happens because corticotropin releasing hormone, or CRH, activates FAR, which is the enzyme that breaks down anandamide. This drop removes AEA's natural inhibitory effect on the HPA axis and allows the stress response to kick in, which contributes to anxiety, reduced neurogenesis, and even anhedonia. But later, as stress hormones like corticosteroid rise, the brain compensates by increasing 2-AG levels, especially in regions like the medial prefrontal cortex and the hypothalamus. And so, this rise in the 2-AG molecule concentration activates those CB1 receptors and helps shut down the HPA axis and its activation, which promotes stress recovery and then also plasticity. And so we've taken a bit of a deep dive there on 2-AG and anandamide, and what we wanted to do was show you the wider family of endocannabinoids that are still being explored and uncovered, such as NADA, PEA, and OEA, and other molecules that also act in this system. And so each of these have very interesting utilities and properties and therapeutic potential as well. PEA, for example, is used in supplementation in autistic people who have a deficit in this endogenous hormone. PEA supplementation has been shown to influence the immune response, reduce neuroinflammation, and improve mitochondrial function and microbiota activity, which means a better quality of life for patients with autism. And this includes the mitigation of many common conditions that also affect people with autism. So, when we talk about the endocannabinoid system, you will encounter a term called endocannabinoid tone, which refers to the functional state of the endocannabinoid system in a given tissue or at a given time. In other words, it is the balance between the production, release, action, and degradation of endocannabinoids, and it can vary from person to person or according to factors such as stress, diet, inflammation, or sleep. We already know that the ECS is more than CB1 and CB2 receptors, but tone is influenced by non-cannabinoid receptors such as the TRPVs, the PPARs, and the G protein coupled receptors as well. Included in this are also the synthesis and degradation enzymes, the The body finally regulates the time of action of the endocannabinoids through these enzymes that are synthesizing and degrading them instantaneously almost, which prevents excessive signaling. The main enzymes to be aware of are the FAR group, which degrade anandamide, the MAGL enzymes, which degrade 2-AG, and the DAGL and PLC phospholipase C, which are the synthesis enzymes. Amongst this group, we've also got serum albumin and other transporters which assist in the transit of endocannabinoids to their destination or active sites, and structures such as ceramides, exosomes, and cholesterol, which modulate membrane fluidity and the accessibility to those cannabinoid receptors. And so, in other words, it is not enough to produce the endocannabinoid, it needs to reach the receptor, perform its function, and then be inactivated just at the right time. And so, this adaptive tissue tone, the balance between these 3 points of production, action, and degradation, are what define this endocannabinoid tone. And this tone is not the same throughout the body by any means. In the brain, it may be more focused on regulating synaptic plasticity, in the intestine, on controlling inflammation and gut motility, or if it's in the skin, it's looking at the homeostasis of, say, the epidermal barrier. And so, therefore, understanding endocannabinoid tone means understanding how dynamic, how adaptive, and deeply involved this system is in the homeostasis of all tissues in the body. And that is precisely why it has become such a promising therapeutic target. One thing to reiterate, the ECS is not just a brain-based system, it's a multifunctional regulator acting across many biological systems. And when it's out of balance, it can contribute to the development or worsening of diseases. And that's why understanding and potentially targeting the ECS has enormous clinical relevance. So this figure here offers a clear visual of the distinct yet overlapping roles of the two cannabinoid receptors. Starting with CB1, we see it is mainly concentrated in the central nervous system where it influences neurological functions. The classic reward, motivation pathways, motor coordination, pain perception, appetite regulation, and energy balances. In contrast, CB2 is most abundant in immune cells and peripheral tissues, so it's playing a critical role in modulating that immune system, reducing that inflammation, and even regulating things like liver fibrosis. Within the brain, CB2 expression is more limited, but becomes active in microglia during, say, diseases or inflammation. So together, these receptors form a bit of a bridge between the nervous system and the immune system, and coordinating responses that maintain the environments in both settings. As research continues, we're now uncovering more about how we can target these receptors, how that could support treatments for a range of conditions from chronic pain through to anxiety and autoimmune disorders as well. If we take a look more specifically now at its functions in the gut, just to move away from neurons briefly, this study here shows how the endocannabinoid system contributes to intestinal homeostasis through multiple interconnected mechanisms. So, firstly, it's strengthening the physical barriers and intestinal epithelium by increasing and assembling tight junction proteins like ZO1 and claudins. This helps maintain the barrier integrity and prevents unwanted intestinal permeability. But secondly, it regulates the secretory function by reducing chloride secretion, which helps control fluid balance, especially relevant to conditions like diarrhea and inflammatory disorders of the gut. This study highlights how the ECS's activation, particularly via CB2 receptors in immune cells, suppresses the release of pro-inflammatory cytokines, helping maintain that anti-inflammatory environment within the gut. And so you can see how it's modulating these different areas that are relevant to the microbiome of the gut. And this involvement and bidirectional relationship with the gut's microbiota too. And so endocannabinoids modulate microbial compositions, while microbial metabolites influence endocannabinoid system activity in return. And so you can see how it's not just mediating one cell tissue, it's balancing the communications between multiple tissues and multiple relevant factors within each of the ecosystems, and particularly in the gut in this instance.